Okay, good morning. So we are now starting with our microbiology review. Okay, so first things first, we have the different important things when it comes to specimen collection and processing. Okay, so I have summarized here the most significant specimens and what are our patient preparation and special instructions when it comes to the collection of these specimens. So first, okay, we have abscesses. That would also include lesions, wound, pustule, and ulcer. So the collection of the specimen here depends whether it is a superficial or a deep abscess. Okay, but prior to the collection, we have to wipe the area with sterile saline or 70% alcohol. And if it is a superficial lesion, okay, or a superficial wound, for example, all you have to do is to swab along the leading edge of the wound. While you are going to aspirate the material from wall or excise the tissue if that is a deep wound or a deep abscess. Okay? And always remember that itong ating aspirate, okay, aspirate is the best specimen for anaerobic culture because usually the organisms that are thriving dito sa mga abscesses, especially yung mga deep abscesses, are anaerobic organisms. And again, our best specimen, okay, our best specimen for the collection for the culture of anaerobic organism is aspirate. Okay, best specimen for anaerobic culture. Okay, next we have the blood or bone marrow aspirate. Okay, so first we are going to disinfect the skin with 70 to 90% ethanol or isopropyl alcohol and then 2% tincture of iodine. So I bet we all know no na ito talaga yung ating combination parate no ng disinfectant when it comes to the collection of specimen for blood culture. Okay, that is always an alcohol iodine combination. Okay, so we are going to draw blood at time of febrile episode, no, or best is before the fever spikes. Okay, that is the best time to collect the specimen for blood culture. And we are going to draw 2 sets from right and left arms, and we must not draw more than 3 sets in a 24-hour period. Now, very important guys, no, yung volume when it comes to blood culture. Okay, and for adults patients, we are going to collect at least 20 ml per set, and that is 1 to 20 ml per set naman for pediatric patients, and that is dependent on the patient's weight. Okay, but always remember that the volume is very important when it comes to blood culture. Okay, so usually no, yung ating mga conventional blood culture systems, okay, yung mga conventional blood culture systems, they are recommending a blood, okay, to culture medium ratio of 1:5 or 1:10. No, sorry, baliktad. No, that is the culture medium to blood ratio. So that is 1:5 or 1:10. No, 1:5 or 1:10. Okay, this is the ratio. Okay, recommended no for blood culture. Okay, mostly by conventional blood culture systems. Okay, and usually no yung set natin, 'di ba, meron siyang dalawang bottles usually per set. So one bottle is incubated aerobically and another bottle is incubated anaerobically. That is for us to be able to detect if whether aerobic or anaerobic din yung pathogen. Okay? So that's it, no? Now, for our blood culture, okay, always remember na meron tayong ginagamit dito na SPS or the sodium polyanethol sulfonate, no? So I just have to mention, okay, yung mga dapat natandaan natin when it comes to the SPS, no? So yung SPS natin, this serves as an anticoagulant. Okay, this serves as an anticoagulant. This is also antiphagocytic. Okay, antiphagocytic and anti-complement. Okay? So of course, no, yung pagiging antiphagocytic niya tsaka pagiging anti-complement niya, that is to make sure, no, that the sodium polyinnitol sulfonate supports the viability of the organism that is to be detected. Okay? Also, okay, yung SPS also removes, okay, it removes certain antibiotics. So parang nagfa-function din siya na parang antibiotic removal device, but only applicable for certain antibiotics such as aminoglycosides. Okay, aminoglycosides, and another is polymyxins. Okay? And another natatandaan natin when it comes to SPS is that it is capable of inhibiting, no? Inhibits the growth of certain organisms. Okay? It inhibits the growth of certain organisms. Concrete example, no, of an organism that is being inhibited by sodium polyinnitol sulfonate is Gardnerella vaginalis. Another example is Streptococcus anaerobius. Okay, Gardnerella vaginalis and Streptococcus anaerobius. Now, always remember that in order for us to be able to remove or to remedy this, no, to remove the inhibitory effect, okay, to remove the inhibitory effect, okay, effect of SPS, okay, we are going to add, okay, 1% gelatin. Okay, so take note, 1% gelatin is the one that is being added in order to remove the inhibitory effect of SPS, no, for the growth of certain organisms. Again, I have mentioned already example of organisms that are inhibited by SPS are Streptococcus anaerobius and Gardnerella vaginalis. Okay? Now, next we have for ONP examination, no? So ONP means ova and parasite examination. Okay, so the collection of specimen here depends if whether the patient is an inpatient or an outpatient. Okay, so you are going to collect, okay, 3 specimens every other day at a minimum for outpatients. Take note, 3 specimens every other day. So it doesn't have to be 3 consecutive daily specimens for outpatients. However, for hospitalized patients or inpatients, you should have a daily specimen collected for 3 days. However, specimens from inpatients hospitalized for more than 3 days should be discouraged. Okay? Now, alam naman natin that there are certain interferences, no, that might affect, no, the examination, no, for the ONP examination, and thus kailangan nating mag-withhold for certain days, no, or kailangan nating mag-ipaliban yung specimen collection for certain days, especially if the patient have taken something, okay, that could interfere with the ONP examination. So for example, if the patient have received antiparasitic compounds, barium, iron, cowpectate, okay, metronidazole, milk of magnesia, Pepto-Bismol, or tetracycline, you have to wait for 7 to 10 days, okay, prior to the collection of specimen. Okay. Next, we have hair, nails, or skin scrapings. No? So remember that these specimens are often utilized for fungal culture. Okay, so how do we prepare the site? The nails or skin should be wiped with 70% alcohol. For hair, okay, if you are going to collect hair, you have to collect hairs with intact shaft. For nails, you are going to send clippings of affected area. For skin, scrape skin at leading edge of the lesion. Okay, so I think I'll just have to mention this because recently, no, yung mga kakapasa lang, no, sa ASCP exam, merong question sa kanila, no, which of the following techniques must be employed for skin scrapings? Ano daw yung mga technique or ano daw yung technique na i-employ or dapat gawin sa isang skin scraping specimen? Okay, I think the choices were acid fast staining, gram staining, then there is calcofluor white in the choices. Okay? So among those that are mentioned, isa lang naman yung technique for fungal examination, and that is calcofluor white. Okay? Alam ko kilalang-kilala natin, no? Kilalang-kilala natin yung tinatawag natin na KOH preparation. However, that was not in the choices. Now, ano itong si calcofluor white? Yes, KOH prep is already adequate when it comes to examination of specimens for fungal presence. However, calcofluor white is best, or this one is recommended, considering that this is a fluorescent technique. So kung meron mang mga fungal elements dun sa specimen, that will fluoresce. Okay? So take note of this calcofluor white in your examinations. Okay? Again, yung tanong daw is, you have a skin scraping specimen, ano daw yung gagawin na technique? No? So the choices again are gram staining, acid fast staining, then there was calcofluor white. So the answer would be calcofluor white kasi siya yung technique for the demonstration of fungal elements. Okay? Next, we have sputum. Have patient brush teeth and then rinse and gargle with water before collection. Okay? And again, we have to collect it from a deep cuff, and of course, meron tayong mga pamamaraan for us to be able to know if whether that specimen is really coming from a deep cuff. Okay? So specimen should be examined for suitability for culture with the use of gram stain. And after doing the gram stain, meron tayong scoring technique na ginagamit dyan. Okay? Yung scoring technique na yon, yan yung tinatawag natin na Bartlett's. Okay? The Bartlett's scoring system. Okay? So sinuscore natin yung presence of neutrophils, yung presence of squamous epithelial cells, yung presence of epithelial cells, yung presence of neutrophils or polymorphonuclear cells. Sinuscore natin yung physical appearance ng sputum, if whether it is bloody, mucoid, okay? And in order for the specimen to be accepted, okay, it must have a score of at least 1 in the Bartlett's scoring system. Okay? Next, we have tissue specimens. So disinfect the skin, of course, and do not allow the specimen to dry out. So moisten with sterile distilled water if the specimen is not bloody. Okay? And lastly, how do we collect urine from an indwelling catheter or a Foley catheter? So you have to disinfect the catheter collection port, then aspirate 5 to 10 ml of urine with needle and syringe from catheter tubing. Okay, now, meron tayong tinatawag na specimen prioritization, no? So, for example, pag sinabi nating level 1, these specimens are the ones that are requiring immediate processing. So, sino-sino yung mga example ng specimens na to? We have blood, okay, blood, CSF, okay, we have other body fluids, okay, other body fluids, and we have brain biopsy. Okay, then for level 2, these are unpreserved, okay, unpreserved specimens. Okay, so examples are feces, urine, sputum, tissue, bone, and wound specimens that are unpreserved. Okay, next, level 3, these are those that requires quantitation. So of course, nandyan na yung ating urine. Okay, urine, then yung tissue specimens din. And lastly, okay, yung mga preserved specimens natin, such as urine, no? Urine and feces that are preserved. Okay, that are preserved or fixed. Okay, and of course, no, yung mga swabs na nasa holding media. Okay? And we also have here the different reasons for specimen rejection. So incomplete information on requisition. And we all know that whatever the information on the requisition, it must match the information that are placed on the specimen label. Okay? Specimen submitted in inappropriate or leaky transport container, inadequate specimen, transport time of greater than 2 hours without preservative. Okay? So always remember no na yung ating transport ng ating specimen, no, the transport of the specimen to the laboratory must be within 30 to 2 hours. Okay? Sorry, 30 minutes to 2 hours. Okay, 30 minutes to 2 hours. Okay, at room temperature. Okay, next is anaerobic culture submitted from site where anaerobes are indigenous, multiple specimens submitted from same source on same day, and direct gram stain of sputum of greater than 25 of less than 25 WBC and greater than 10 epithelial cells per LPF. Kasi guys, pag konti yung WBC and marami yung kanyang epithelial cells, that means that the specimen is mainly saliva lang, no? So that is also a reason for what they call this, specimen rejection. Okay? Now let us go to biosafety, no? So from time to time tinatanong pa rin ito in my exam. Actually, I have a question about a pathogen that requires biosafety level 2 handling, no? May ganun akong tanong sa aking exam, no? So kailangan pa rin natin tong aralin. So first, we have our personal protective equipments. So that includes gloves, laboratory coats, masks, respirators, face shields, and safety glasses. And always remember that these PPEs must never be worn outside the laboratory. Okay? Then we have universal precautions. Universal precautions is applied when handling all patient samples. So we are regarding that all patient samples are infectious. So it is assumed again that a specimen contains biological agents that correlate with at least a biosafety level 2. Okay? So that means no, pag sinabi nating biosafety level 2, these are those that are common agents of infectious disease that are not airborne. Okay? So guys, take note natin yung biosafety levels no ng ating mga organism. So when we say biosafety level 1, these are organisms with no known infectious potential. Example, yung mga contaminants natin such as Mycobacterium bourdainei and Bacillus subtilis. Pag sinabi nating level 2, most human specimens are containing biosafety level 2 agents. So these are common agents of infectious disease that are not airborne. So that includes Salmonella, Shigella, Streptococcus pneumoniae, HIV, Bacillus anthracis, and Yersinia pestis. Then we have biosafety level 3. These are those that cause serious disease and are airborne. So that includes Mycobacterium tuberculosis, Francisella tularensis, Brucella, and we have the systemic fungi. Okay, so in my exam, sorry, no, I mentioned earlier that it's level 2, but it's actually level 3. Yung question sa exam ko is which of the following organisms require a biosafety level 3 handling? Parang ganon. And in the choices, I could vividly recall na dun si Coccidioides immitis. And I'm sure that that is the answer. Okay? Kasi si Coccidioides, no, Dioides immitis, this is one of the fungal pathogens no that are really presenting danger sa mga laboratory workers, no, considering that its mode of transmission is inhalation, no, so airborne siya, no, kasama niya actually no si Histoplasma capsulatum. Okay, so sila yung mga fungi no na kailangan natin talagang i-handle ng maayos kasi pag na-inhale natin yung kanilang mga spores, no, we will be infected. Okay? So Coccidioides immitis and Histoplasma capsulatum. Actually, there is an old recall question na parang tinanong lang on what is the mode of transmission of Histoplasma capsulatum, and that is inhalation. Okay? Then we have level 4 pathogens. Yung mga level 4 pathogens naman, these are those that are causing serious disease that are often untreatable or wala pang known treatment, and they are also airborne. So that includes arbovirus, arenavirus, filovirus, and the smallpox virus. Okay? Then we also have our biosafety cabinets. Okay? So kailangan natin ito when handling specimens. Okay, and in doing our culture, diba? So biosafety cabinets should be used properly to process all specimens. We decontaminate interior workspaces with the use of 70% ethanol. Do not place items on front intake frill or the rear wall or floor exhaust grills kung saan lumalabas yung hangin or kung saan lumalabas-pasok yung hangin. Hindi tayo pwedeng maglagay ng items doon. Okay? So allow the blower to run 15 to 30 minutes before and 30 minutes after the use, and turn on UV light to disinfect interior of cabinet when not in use. Okay? So usually daw, di ba, meron tayong parang sash opening, no, sa ating biosafety cabinet. So the sash opening is usually 8 to 10 inches. So usually when you are sitting in front of a biosafety cabinet, yung sash daw is in the level of your axilla or your armpit, no? So the opening is approximately 8 to 10 inches, no? Kasi pag masyadong malaki, no, if the opening is too wide, alam naman natin na merong alarm system, no, yung ating biosafety cabinets, and usually nag-a-alarm iyon if the opening, no, the sash opening is too wide. Okay? And syempre nako-compromise yung integrity ng environment sa loob if the sash opening is too wide. Okay? So meron tayong tatlong classes of biosafety cabinets. Pag sinabi natin class 1, that is the open front with negative pressure, and class 1 biosafety cabinets are only sterilizing the air that is to be exhausted. Take note, class 1 biosafety cabinets are only sterilizing the air that is to be exhausted. Now, yung ating class 2 biosafety cabinets, this is the most common na ginagamit na biosafety cabinet and is also known as the laminar flow biosafety cabinet. So this type of biosafety cabinet is sterilizing the circulating air and the air to be exhausted. Okay? So pag class 1, sinisterilize niya lang yung lumalabas, kaya 1. Yung lumalabas lang na air, yung sinisterilize niya. Yung class 2, sinisterilize niya yung nagsi-circulate tsaka yung lumalabas na air. Yan yung palatandaan ko eh. Kaya 2, dalawa. Dalawang uri ng air yung sinisterilize niya. Yung nagsi-circulate tsaka yung lumalabas. Now, for the class 2 biosafety cabinets, there are 2 types. We have class 2A and class 2B. Pag sinabi natin class 2A, ito yung tinatawag natin na self-contained. Okay? Self-contained biosafety cabinet wherein 70% of air is recirculated. Take note, 70% of air is recirculated. Okay? Then meron tayong class 2B. Pag sinabi natin class 2B naman, 30% naman ng air yung nare-recirculate. Okay? 30% of air is recirculated, and oftentimes itong class 2B biosafety cabinet natin are the ones that are used for radioisotopes. Okay, radioisotopes, toxic chemicals, or carcinogens. Okay, then finally, we have our class 3 biosafety cabinet. So sa class 3 biosafety cabinet, the system is entirely closed. And usually, ito yung may rubber gloves for you to be able to maneuver or to do the things to be done inside the biosafety cabinet. So may gloves yun na naka-embed na or naka-install na doon. And of course, yung class 3 biosafety cabinet gives the highest level of safety. Okay? So sterilized air entering, circulating, and to be exhausted are the ones that are being sterilized. So tatlo yung uri ng air na sinisterilize ng class 3 biosafety cabinet. The entering air, the circulating air, and the air that is to be exhausted. No? So yan yung tatandaan natin dyan. Okay? Then, we have here the CDC classification of bioterrorism agents. Guys, we really have to memorize this kasi may recall question na tinatanong lang talaga, which of the following is considered as a category A? bioterrorism agent. Ganun lang, no? So sa choices naglalagay sila ng combination ng mga agents, no? So you have to choose there kung sino yung category A. So syempre pwedeng maiba yung tanong, and let's say tatanungin nila yung category B or category C. So wala talaga tayong choice dito but to really memorize kung sino-sino yung mga members under this categories, no? So pag sinabi natin category A, ito yung mga highly infectious na mga bioterrorism agents. And of course, since they are highly infectious, they are the ones that are given with highest, okay, priority. Okay, the one given with highest priority. So that includes variola, hemorrhagic fever viruses, Bacillus anthracis, Yersinia pestis, Clostridium botulinum, and Francisella tularensis. Now for category B, we have moderate morbidity and low mortality. No? So these are agents that are causing moderate morbidity and low mortality. So we have encephalitis viruses, Brucella species, Burkholderia malae, Burkholderia pseudomalae, Chlamydia psittaci, Coxella burnetii, okay, Rickettsia prowazekii, Clostridium perfringens, Staphylococcus aureus, certain foodborne bacteria and waterborne bacteria. And for category C, no, so syempre sila yung may third priority. These are those emerging pathogens such as influenza virus, Nipah, hantavirus, rabies virus, yellow fever virus, drug-resistant tuberculosis, and Rickettsia connori. Okay? So again, memorize natin itong CDC classification of bioterrorism agents. Okay, so let us now proceed to the direct microscopic examination. Okay, so first, of course, we have gram staining. Okay, so yung gram staining, this is routinely performed on certain specimens or certain specimen types, especially those collected from normally sterile sites. Okay, itong gram staining is our guide, no? For us to be able to know, of course, the morphology of the organism. Kasi sometimes, no, yung morphology, not sometimes, but morphology is really an important parameter for us to be able, important parameter to be used, no, when it comes to species identification. Okay? So, of course, no, yung reagents natin for gram staining. So, we have our primary stain. We have crystal violet, no? We have our crystal violet. Then the mordant is grams iodine. Okay, grams iodine. Then we have acetone alcohol as our decolorizer. Okay, and we have safranin as our secondary stain or our counter stain. And always remember that the most significant step, no, the most significant step when it comes to gram staining is the decolorization step, because dito guys first nagkakaroon ng difference or ng difference between a gram positive and a gram negative organism. Because if you would notice from the first, no, sa pag-apply ng crystal violet tsaka pag-apply natin ng grams iodine, basically, no, the appearance of the gram pos and gram neg organisms are the same. But the first step wherein we are able to differentiate them is in the decolorization step, because, no, yung ating mga gram positive organism are not decolorized, okay, whereas gram negative organisms are not able to retain the primary stain. Okay? So, however, sometimes, no, organisms can appear as falsely gram negative or falsely gram positive, no? So ito guys yung mga reason for an organism to appear falsely gram negative, no? So pag sinabi nating falsely gram negative, the organism is gram positive but is appearing red or pink, no? So the organism is gram positive, no? The organism is gram positive but is colored, no? But is colored red or pink. Okay? So falsely gram negative siya, no? So if you over decolorize, okay, you miss the iodine, or if you make a gram stain from old or dying colonies. Now for falsely gram positive naman, these are organisms that are gram negative, no, gram negative, but is colored blue or purple. Okay, colored blue or purple. So under decolorization, and if you have made a thick smear. Okay? Now let's go to acid-fast staining. Oh, by the way, no, ano ba yung nagpapagrams, ano ba yung responsible for the gram positivity of organism? Okay? So remember na yung substance that is responsible for the gram positivity of organisms is because it contains or it has a thick peptidoglycan layer. Okay? Meron siyang thick peptidoglycan layer. And this layer is also present sa mga gram-negative organisms natin. However, it is just thin, no? Yung mga gram-negative organisms natin, they only have a thin, no, thin peptidoglycan layer. Okay? And yung main component talaga ng mga outer membrane nila is yung kanilang lipo, okay? Meron silang lipopolysaccharide. Yan. Okay? Next, we have our acid-fast staining. So for acid-fast staining naman, usually this is utilized for mycobacteria. Okay? Yung acid-fast staining. Yung mga mycobacteria natin, basically, they have a structure similar to that of gram-positive. They have a cell wall structure similar to that of gram-positive organism. However, because their cell wall also contains mycolic acid, this substance repels crystal violet. Okay? So again, lagay ko dito they have a similar structure. They have a cell wall structure of gram-positive organisms. Okay, so technically they are gram-positive. However, no, because of mycolic acid, okay, okay, crystal violet, which is the primary stain for gram stain, no, crystal violet is repelled. Yan. Okay? So we have, actually marami tayong uri, no, ng acid-fast staining because we have a lot of modifications of it. But I've enlisted here the main ones. We have the Ziehl-Neelsen technique. Okay, so we have the Ziehl-Neelsen technique. So for the Ziehl-Neelsen technique, we have carbulfoxin as our primary stain. The more that is heat, kaya siya tinawag na hot technique or the hot acid-fast technique. And yung ginagamit natin na decolorizer is 3% acid alcohol and the counter stain is methylene blue. Of course, acid-fast organisms will take up the carbulfoxin appearing red, whereas non-acid-fast organisms will be colored blue because of the counter stain. Okay, for the Kinyon technique, still the primary stain is carbulfoxin, but this time we are not using heat as a mordant. Instead, we are using detergents, okay, such as phenol and tergitol, and the purpose of these detergents is to be able to penetrate the hydrophobic barrier. The hydrophobic barrier that we are referring to is the mycolic acid. So in order for us to be able to penetrate the in order for the carbulfoxin rather to be able to penetrate the hydrophobic barrier of an acid-fast organism, gumagamit ng phenol or tergitol. Okay? Still, the decolorizer is 3% acid alcohol. And for the counter stain, we could use malachite green or methylene blue. No? Pwede din yung malachite green, pwede din yung methylene blue. Okay? So again, hindi tayo gumagamit dito ng heat, kaya siya tinawag na cold technique. And this is the type or this is the modification of acid-fast staining na ginagamit natin for tissue specimens. Okay? So still acid-fast organisms will appear red, whereas non-acid-fast organism will appear blue or green depending on the counter stain being used. Okay? Now, for our uramine rhodamine stain, this is the most sensitive among all acid-fast staining, obviously, kasi fluorescent technique siya. So dito, gumagamit pa rin tayo ng acid alcohol as a decolorizer, however, at a much lower concentration, which is 0.5%. Then, guys, take note of potassium permanganate here. That is our counter stain, or yung lumalabas sa ASCP exam, ano daw yung purpose ng potassium permanganate when it comes to acid-fast staining? That serves as a quenching agent. Okay, it is serving as a quenching agent. So of course, since this is a fluorescent technique, acid-fast organisms will fluoresce, whereas non-acid-fast organisms won't. Okay, so they are non-fluorescent, okay, sa technique na ito. Okay? Then we have the modified acid-fast staining technique, or the FITE acid-fast technique, which is utilized for weakly acid-fast organisms. Okay? So example of weak acid-fast organisms are Nocardia, Cryptosporidium, Cystoisospora belli, which is formerly known as Isospora belli, Cyclospora, Cercocystis, and Legionella micdodae. So always remember na yung pinagkaiba nitong FITE acid-fast staining technique, or the modified acid-fast staining technique, from those that are utilized kanina, yung Zillnilson and tsaka Quinion, because this one is making use of a weaker decolorizing agent. No? Gumagamit siya ng weaker decolorizing agent. So that's the difference of the modified acid-fast stain. Okay? And lastly, we have acridine orange. So acridine orange naman is a stain for the DNA, no? It detects nucleic acid, no, yung acridine orange. So it could be used, no, for smears prepared from blood and body fluids where organisms are in low concentration. Because itong acridine orange, sensitive din siya because this is also a fluorescent technique, no? Sa katunayan, ito guys yung technique na ginagamit natin sa QBC, no? Yung quantitative Buffy coat, no, for malarial parasites, no? Yung quantitative Buffy coat for malarial parasites. So considering its sensitivity, magandang gamitin, no, itong acridine orange if there is a low concentration of the organism. So take note that this acridine orange is can be used, no, for the detection of bacteria and yeasts. Okay? Bacteria and yeasts. Okay? So now let us proceed to the different media for routine bacteriology. So yung mga media natin, guys, they are being classified into several types. Okay? So unahin muna natin, no, yung mga types of culture media natin. Okay, dito tayo. Okay? So we have types. Okay, of culture media. Okay? So first, okay, yung mga tinatawag natin, guys, na non-selective. Okay? Non-selective, or minsan they are referred to as general isolation media. So pag non-selective, that means this type of media, okay, adequately supports growth of most organisms. Adequately supports growth of most organisms. Okay? So, concrete example natin dito is yung ating TSA, no? The tryptic soy agar. Okay? Then, we also have our enrich, no? Meron tayong tinatawag na mga enrich media. Okay? Pag sinabi naman natin na enrich media, meron siyang, it contains growth enhancers. Sorry. It contains growth enhancers, no? So, example ng mga growth enhancers that can be incorporated to this media includes 5% sheep's blood. Okay? 5% sheep's blood. Pwede tayong maglagay ng mga vitamins. Okay? Yan. So that makes your blood agar plate as an example of an enriched media. Okay? Then we also have our selective. Okay? Selective media. So yung mga selective media naman, from the name itself, selective, it selects only for growth, no? It selects for growth. Okay? Of a group of organisms. Okay? And nagiging selective siya, no? By adding, okay, by adding inhibitory substances. Okay? Example ng mga inhibitory substance na pwede nating ilagay, dyes, we have alcohols, okay, alcohols, acids, okay, or antimicrobials, no? So ito yung mga pwede nating i-incorporate, okay, sa isang selective media in order to prevent the growth of a certain group of organism and will only allow growth or select for growth of a certain group of organism. Okay? And example, no, example ng mga selective media natin, no, syempre yung McConkey agar natin, no? Because your McConkey agar can only support the growth of facultative, okay? Gram-negative organisms, no? Gram-negative. Okay? We also have yung tinatawag natin na CNA, no? CNA is colistin nalidixic acid agar, no? Colistin nalidixic acid agar, which only supports the growth of gram-positive, okay, gram-positive cocci and bacilli. And lastly, meron tayong tinatawag na differential media. So yung ating mga differential media, para din silang mga selective. Actually guys, no, sa katunayan, most differential media are also selective media. Okay? So pag sinabi naman natin na differential, it supports growth of a group of organism. Okay? Of a group or groups of bacteria and at the same time differentiates. Okay, differentiates within the group. Okay, so earlier I mentioned that most differential media are also selective media. So gagamitin natin na example si McConkey ulit. Kasi si McConkey only is selective because it does not allow gram-positive organism to grow, no, sa McConkey agar. And at the same time, it is differential because we are able to differentiate between lactose fermenters and non-lactose fermenters with the use of McConkey agar. Okay? So yan po na yung mga examples ng ating culture, yung mga types rather ng ating culture media. Okay? So I have here an example or a list of the commonly used non-selective media. So pag sinabi nating non-selective ulit, it supports growth of most organisms, no? So for example, we have sheep blood agar. Okay, it supports the growth of most bacteria. However, meron siyang mga exceptions. No, so sino-sino yung mga organism na hindi tumutubo sa ating sheep's blood agar or sa ating SBA or SBAP? We have Neisseria gonorrhea. Okay, we have Haemophilus influenzae. And we also have Legionella. Okay? Then we also have chocolate agar. This is for the cultivation and isolation of fastidious, no? Fastidious, mga medyo maaarte na organism. So example ng mga fastidious organisms, of course, yung ating Neisseria species. Okay? Neisseria species. We have Haemophilus species. Yan. But it does not, no? Yung chocolate agar, take note, does not support the growth of Legionella because we have a specific agar for Legionella, and that is your BCYE, your buffered charcoal yeast extract. Okay? Lumabas na yan sa exam. This one contains cysteine and iron supplementation, which is needed, no, for the growth of Legionella species. And it also contains activated charcoal, which helps bind and sequester growth inhibitors that might be present in the media. Then we also have our Mueller-Hinton agar, which is utilized for antimicrobial susceptibility testing of many common bacteria. And we also have thioglycolate broth, which is for the cultivation of bacteria, including microaerophils and obligate anaerobes. So itong thioglycolate broth, it supports the growth of several organisms, and at the same time, we are able to classify these organisms based on their oxygen requirement. Kasi nalalaman natin kung ano yung kanyang oxygen requirement based sa location sa thioglycolate kung saan siya nag-grow. So for example, if the organism is an obligate anaerobe, you might be seeing that in the bottom of the thioglycolate broth kasi takot siya sa oxygen. Usually yung mga microaerophil, they are in the subsurface. Kasi yung nandun sa surface are the obligate aerobes. Okay? Always remember that the oxygen tension decreases towards the bottom of the thioglycolate broth. No? So it permits again growth of several organisms, and at the same time we are able to classify these organisms according to their oxygen requirement. Okay? Then we also have here commonly used selective media. So we have McConkey. It contains bile salts and crystal violet. We have eosin methylene blue. It contains aniline dyes. We have Campylobacter blood agar, which contains antibiotics such as cephalotin, vancomycin, trimethoprim, amphotericin B, and polymyxin B. Okay, so just take note, no? Just take note kung ano yung mga utilization ng mga selective media na ito, no? I'll just have to point out, no, yung ating Salmonella shigella agar, kasi yung pangalan niya is Salmonella shigella agar, no? It contains bile salts, sodium citrate, and brilliant green, which is utilized for the recovery of Salmonella and Shigella. However, these Salmonella shigella agar may inhibit Shigella. Okay? This may inhibit Shigella. Okay? And thus, it is not recommended for its primary isolation. Okay? Not recommended for primary isolation. Okay? Alam na natin yan. Okay? Yung ating SSA agar. Because again, at times, no, it may inhibit, no, the growth of Shigella. Okay? Then, we have CIN, no? The Cifsuludin, Ergasan Novobiocin agar. Take note, no? This is for the recovery of Yersinia species, no? That is also ASNA in the ASCP exam. Okay? Then we have the anaerobic colistin nalidixic agar. Okay? Then this is utilized for the recovery of anaerobic streptococci. Then we also have the limb broth. Okay, the limb broth is basically containing also colistin and nalidixic acid. Okay, pareho siya ng content. No, meron siyang colistin, meron siyang nalidixic acid. It's just that it is a liquid media. Liquid lang siya. Itong isa, this is agar. Okay, or solid. Okay? Then we have the Regan Low. This is utilized for the recovery of Bordetella pertussis and Bordetella parapertussis. So it contains antimicrobials to be able to prevent the growth of unwanted organisms. And it contains antimicrobials. No? Concrete example is yung cefalexin. No? Baka itanong ano daw yung antibiotic na laman ng Regan Low medium, that is cefalexin. Then we also have our Thayer-Martin medium, which is for the recovery of Neisseria species. So it contains vancomycin. Okay. And the vancomycin here is to prevent the growth of gram-positive organism. Then meron din siyang colistin. Okay, colistin. The purpose of colistin is to prevent the growth of gram-negative organism. Then meron din siyang nystatin. Okay, nystatin. The purpose of nystatin is to prevent the growth of fungi. Then meron din siyang trimethoprim lactate, no, or the SXT, no, the trimethoprim. The purpose of this is to prevent the what do you call this? To prevent the swarming of Proteus. Sa katunayan, there are two types, diba, of Thayer-Martin. We have Thayer-Martin and the modified Thayer-Martin. The difference between the two is that only the modified Thayer-Martin has SXT. Siya lang yung may trimethoprim. Yung Thayer-Martin na basic, wala siyang trimethoprim. Okay? So, yeah. Then we have commonly used differential media. So oftentimes para ito guys, no, for the differentiation of lactose and non-lactose fermenters. So I think here it's important for us to note on what is the color of lactose and non-lactose fermenters sa mga media na ito. Okay, so first we have our McConkey. Again, this is a differential and a selective media at the same time. This is used for the differentiation of lactose fermenting and non-lactose fermenting enterics. Lactose fermenters here will exhibit pink, okay, pink or red colonies. Okay, whereas non-lactose fermenters will have translucent, okay, translucent colonies. So usually yung mga non-fermenters, they are exhibiting translucent colonies. That's true across all of these agar, no? So pakilagyan na diyan sa mga non-lactose fermenters, translucent yung kanilang colonies. Yeah. Explain ko yan mamaya, yung sa TCBS. Okay? Now, balik tayo doon sa ating eosin methylene blue. We have EMB, okay, or eosin methylene blue. So this is again for the differentiation between lactose and non-lactose fermenters. So pag lactose fermenter ka, you will be exhibiting purple-black, okay, with green metallic sheen. Okay? Then we have hectone enteric agar. Yung mga lactose fermenters tsaka sucrose fermenters, they will have yellow, okay, or orange, okay, colonies. Then pwede ding ma-exhibit dito yung H2S production na nagkakaroon ng black coloration. Okay? Then we have SSA, Salmonella shigella agar. Lactose fermenters will have pink or red colonies. Okay, non-lactose fermenters, of course, will have translucent colonies, and H2S producers will have black coloration. Okay, then we have TCBS. So usually para to sa mga Vibrio. Okay, so yung mga sucrose fermenters will have yellow colonies, whereas non-sucrose fermenters will have green or translucent colonies. Then we also have CIN. CIN is again Cefsulodin ergazan novobiocin. So this is for the differentiation of mannitol fermenting and non-mannitol fermenting Yersinia species. So yung mga mannitol fermenting, they will have an characteristic bullseye colonies. Okay? Bullseye colonies. So pag sinabi natin bullseye colonies, the colonies are colorless but with red center. Kaya siya bullseye, no? Colorless with red center. Okay? And lastly, we have our motility test agar. So since this is a motility test agar, take note always that this has to be a semi-solid media. No? This has to be a semi-solid media. So used, no, for the differentiation of motile and non-motile bacteria, malalaman natin that the organism is motile if there is a growth. Okay, if there is growth away from the line of inoculation. Okay, growth away from the line of inoculation. So usually, because the organism is growing away from the line of inoculation, it tends to cloud, no? It is resulting to clouding of the agar. Okay? Clouding of the agar. Then pag non-motile, obviously it will stay, okay? Stay within the stab, okay? Or the site of inoculation, and of course the agar here is clear. Okay? So yan po no yung mga differential media natin. Okay, so we are now down to our last topic for this session. We have antimicrobial susceptibility and resistance. And this is one of the high yield topics no in the ASCP exam when it comes to micropara. Sa katunayan, during my exam, I have a lot of questions. I cannot recall any question about identification. Parang lahat is something with antibiotics talaga, no? So kailangan natin talaga tong aralin, no, yung antimicrobial susceptibility and resistance. So first, of course, no, yung mga classes of antimicrobial agents natin, we have to be familiar with them, kung sino yung i-inhibit ng cell wall synthesis, sino yung nagdi-disrupt ng cell membrane, sino yung nag-i-inhibit ng protein synthesis, the one that inhibits nucleic acid synthesis, the one that inhibits metabolic processes, antimicrobacterial agents, and antifungal agents. Okay, so I will only focus on a certain selected antibiotics here. So first, we have our antibiotics that inhibit cell wall synthesis. So we have here penicillin, cephalosporin, carbapenem, monobactams, glycopeptides, and lipoglycopeptide. Now, I just would like to expound on cephalosporins, that we have several generations of cephalosporin. Okay? So usually, no, yung first generation natin, okay, ano ba tawag dito? Generations, no, of cephalosporins. Okay, so we have first generation, we have cefazolin, and cephalexin. For the second generation, we have cefuroxime. We have cefaclor and cefoxitin. For the third generation, we have ceftazidime, ceftriaxone, and cefixime. Okay. Then we have, for the fourth gen, we have cefepime. And for the fifth gen, we have ceftobiprole. Okay. Also, I would like you to take note also of the type of penicillins that are used for penicillinase resistant organisms, no? So, penicillinase resistant na mga penicillin. So, that means if the organism is a penicillinase producing organism, these are the type of penicillins na binibigay sa kanila. Okay, so we have methicillin, or these are the antibiotics na pwedeng maging effective for them. We have methicillin, we have nafcillin, and we have oxacillin. So again, these antibiotics are penicillinase resistant. So that means they would be effective for penicillinase producing organisms. Okay? Okay. Next, we have, let's go to the different methods of susceptibility testing. So first, we have the disc agar diffusion, or mas kilala natin to sa tawag na Kirby-Bauer diffusion technique. So take note of these parameters. Natatandaan natin, we have the standard inoculum. So the standard inoculum concentration, kumbaga, should be 1.5. Okay, 1.5 times 10 to the 8 colony forming units per ml. And for us to be able to achieve this, that's why kino-compare natin siya with our standard for the inoculum, which is our 0.5 McFarland standard. Okay, then the standard media is, of course, we have the Mueller. Okay, we have the Mueller-Hinton agar. Okay? So usually gumagamit tayo dito ng 150. Usually 150 mm plates yung ginagamit natin, but we know na meron tayong 100 mm plates na nag-e-exist. So usually 150 mm plates, then 4 mm yung depth or yung thickness, and 7.2 to 7.4 yung pH. Okay? Next, for the incubation period, that is 35 degrees Celsius for 18 to 24 hours. No? Kasi usually meron tayong 16 to 18 hours incubation. Then there are those that requires up to 24 hours incubation. Okay? Of course, you have to take note of the divalent cations, which particularly important when we are testing Pseudomonas species against aminoglycosides. So, sino ba yung mga divalent cations na ito? We have calcium, okay, and magnesium. Okay, so to read the zone of inhibition, the plate should be held 2 to 3 inches above a black surface using reflected light, except for enterococci with vancomycin and staphylococci with oxacillin. Kasi dito sa enterococci that is tested with vancomycin and staphylococci that is tested with oxacillin, yung ginagamit natin dito is the transmitted light. Okay, and if there is swarming over a zone of inhibition, it should be ignored. Okay, now, ano ba yung mga possible causes that the organism might appear falsely resistant and might appear falsely susceptible? So pag sinabi nating falsely resistant, that means that there is a narrow, okay, there is a narrow zone of inhibition. Ako guys, yung palatandaan ko dito, nilalagyan ko parati yan ng smiley face, yung falsely resistant. Because these are the situations, no, or the instances wherein the organism is liamado, no? Kumbaga, if the organism is in an advantage, okay? So they are appearing to be falsely resistant. Kumbaga, nananalo sila. Kaya nilalagyan ko yan parati ng smiley face for the falsely resistant. So ano ba ito, no? First, we have too heavy. Okay, too heavy inoculum. Siyempre, mas masaya kung marami. No? So, kung marami sila, mas masaya, di ba? Too thick agar, di ba? Mas maraming pagkain. Okay? Then, alam naman natin na gusto-gusto nila yung moisture. So, too much moisture. And number 4, improperly stored discs. Okay? Then for our falsely susceptible, lagyan naman natin ng sad face kasi ibig sabihin, wide naman dito yung zone of inhibition. Okay? Ito naman yung mga instances wherein natatalo yung organism. Okay? So, if there is too light inoculum, okay? If the agar is too thin, and if the agar is dry, of course. Okay? So, always remember pala no yung 15 minutes rule natin when when we are doing the agar diffusion technique. So dapat 15 minutes pagkatapos mong gawin yung inoculum, you have to uh, inoculate it in the agar already. No? So 15 minutes, within 15 minutes from the preparation of the inoculum, you have to inoculate that onto your Mueller-Hinton agar. Then you have to wait for 15 minutes before placing the antibiotics. Then you have to set it aside for 15 minutes before placing inside the incubator. No? So 15 minutes natapos mo yung, gumawa ka ng inoculum, within 15 minutes, inoculate mo siya sa agar. Within 15 minutes, maglagay ka ng antibiotics. Within 15 minutes, lagay mo siya dun sa incubator in an upside down position, of course. Okay? Now, take note lang natin, guys, that this agar diffusion technique is not useful for anaerobes and slow-growing organism. Okay? So, take note that this cannot be used, okay? Cannot be used for anaerobes and slow-growing organisms. Okay? Yeah. Sorry, organisms. Now let us proceed to another method. We have the e-test. Okay? So we are using here a strip, no? Nakikita naman dyan sa ating illustration, no? Sa info, sa picture natin dyan. We are using a strip placed on an MH agar plate that has been inoculated with the organism. And the strip contains gradually increasing concentrations of a specific antibiotic. Now, the result is read as a minimum inhibitory concentration where the zone of inhibition intersects with the strip. So, dito, whatever the concentration there, yan yung kanyang minimum inhibitory concentration. Now, tatandaan natin na sa ating e-test, this can be used for anaerobes and fastidious organism. No? This can be used and fastidious organisms. Okay. Next, we also have the Malditov MS. So Malditov means matrix-assisted laser desorption ionization time of flight mass spectrometry. So nakalagay siya dito, guys, sa methods of susceptibility because today, may research that kumbaga ine-explore pa yung possible use of the Malditov MS in rapid antimicrobial susceptibility testing, but it is not yet officially employed for rapid AST. Kumbaga, ini-explore pa lang. I am emphasizing this because nandyan yan sa ASCP exam. Now, which of the following, kumbaga daw, yung advantage of the Malditov? And nandun sa choices, guys, yung rapid antimicrobial susceptibility. So, we cannot answer that yet because, kumbaga, nasa research pa lang siya. Your Malditov MS assay is useful for rapid identification of pathogens, but is yet to be used for rapid antimicrobial susceptibility testing. So this is an emerging technology that identifies infectious pathogens by defining unique protein signatures of the organism. Guys, take note protein. Okay, lumabas yan sa exam ko. Ano daw yung dinidetect ng Malditov? Malditov is proteomics. So protein yung dinidetect natin dito. So the procedure, an isolated colony is ionized. Guys, lumabas to sa student ko na nag-take last week lang ng exam. Ano daw yung process na involved in Malditov? Yung mga choices niya, deamination, ionization, mga ganyan. So take note the process involved is ionization. Okay, resulting in the vaporization of the proteins. Now, the proteins are separated based on size and charge in an electric field resulting in a unique spectral signature. Then this spectral signature is being compared with those existing spectral signature for an identified organism. Okay, so yan po si Malditov. Okay? Now, let's go to bacterial resistant mechanisms. So ito guys, yung mga bacterial resistance mechanisms natin. We have organisms that are naturally or intrinsically resistant. So example ng mga organism na naturally resistant to a certain antibiotic, we have Enterococci. They are naturally resistant to cephalosporins. Okay. Then we also have Klebsiella pneumoniae. Okay, that is naturally resistant to ampicillin. Okay, now watch out for this question in the ASCP exam. Okay, because there is a parang case study wherein na-identify daw na yung organism is Klebsiella pneumoniae. Then sa pag-perform ng antimicrobial susceptibility testing, it was found out that the organism was susceptible to ampicillin. Ano daw yung gagawin? Tapos yung sagot doon, because that is actually a lifted question from BOC. Yung sagot doon is you have to recheck because Klebsiella pneumoniae is not susceptible to ampicillin. Okay? So baka may mali no sa procedure. Okay? Then other bacterial resistant mechanisms, we have chromosomal mutation, target site changes, reduced permeability of the drug, enzyme production, plasmid production, and decreased permeability or uptake or increased efflux of the antibiotic. Okay? So usually this is because of either there is a change, okay? There is a change in the character of foreign channels. Change in the character of porine channels, or this could be due to an increased ability of organisms to pump out cell contents. Kaya lumalakas yung efflux or yung paglabas ng antibiotic from the organism, kaya hindi siya na-affect ng mabuti, kumbaga. Okay? So, I just would like to mention about our ESBLs, tsaka yung MAC-A. Yung ESBLs, that means extended spectrum beta-lactamase. So, beta-lactamase is produced by certain organisms such as Escherichia coli, Klebsiella species, Proteus mirabilis, and other members of the Enterobacteriaceae. So, itong beta-lactamase, it hydrolyze and render inactive penicillin, cephalosporins, and aztreonam. So that means if the organism is an ESBL, these are the antibiotics that must not be given to them kasi wala silang silbi, kumbaga. Then we also have the MEC-A. The MEC-A is the gene responsible for methicillin resistance. So this is found in bacterial cell walls, and the most commonly known carrier of this gene is the methicillin-resistant Staphylococcus aureus. Okay? So pwede tayong tanungin ano daw yung best method or the gold standard for the detection of methicillin-resistant. That would be detection of MEC-A gene through PCR. That would be the gold standard for the detection of methicillin resistance. Okay? Now, kung meron tayong mga method for antimicrobial susceptibility, of course, we also have our methods for testing resistance. Okay? So, first, we have the beta-lactamase enzyme detection. So, this is performed on isolated colonies or on isolates, on isolates of Staphylococcus, Haemophilus, Neisseria gonorrhea, and Moraxella catarrhalis. So, the methods that are used, pwedeng gumamit ng chromogenic nitrosophene method, which is a common method, and parating tandaan dito that the best substrate is nitrosophene, nitrosophene yung best substrate dito. Okay? So the positive result for this is pink. Okay? Kasi yung nitrosophene originally it is yellow or colorless. And when the organism is a beta-lactamase organism, it breaks the nitrosyphene beta-lactam ring, turning it into pink. Okay? Then we also have the iodometric method. Okay? So the positive result here is no color change. Okay, no color change. So in the iodometric method, gumagamit tayo dito ng starch solution dried on a filter paper. No? So meron tayong filter paper, tapos nilalagyan natin siya ng starch solution, tapos pinapagay natin siya dito. Then naglalagay tayo ng iodine. Tama? So supposed to be, if there is starch in there, it will be colored blue by iodine. But if the organism is a beta-lactamase organism, the iodine is being neutralized. Thus, there is no color change. Kasi kumbaga, yung iodine that is supposed to give blue color to starch was neutralized by beta-lactamase. Okay? And lastly, we have the acidic method. The positive color for the acidic method is yellow. Okay? So, gumagamit tayo dito ng phenol red indicator. Okay? Next, we have the MRSA detection. So we are using certain antibiotics for this. So we have cefoxitin, which predicts the presence of MECA gene. So usually pag cefoxitin, gumagamit tayo ng 30 microgram disc. No, pakitandaan itong mga concentration ng mga disc na ginagamit because I had one in my ASCP exam. Okay? So yung cefoxitin, this is better, okay, better than oxacillin. Okay, better than oxacillin at inducing the expression of MECA gene. Yeah. Okay? Then we also have oxacillin. Pag oxacillin, gumagamit tayo ng 6 microgram disc. Okay? Then this is used on MH agar with 4% NaCl that is incubated at 33 to 35 degrees Celsius in ambient air up to 48 hours. Okay, and of course, our reference method, our molecular method, wherein dinidetect talaga natin yung presence ng MECA gene. Okay, so that is for the MRSA detection. Then we also have the D-zone test. Okay, so the D-zone test is a test for inducible. Ito yung lumalabas sa exam. Para saan daw yung D-zone test? It is a test for inducible clindamycin resistance. So this is performed on some erythromycin-resistant organisms. Kasi yung mga erythromycin-resistant organisms, they may also develop clindamycin resistance whenever they are exposed to it. So the positive result is flattening of the clindamycin zone of inhibition adjacent to erythromycin. So dito ito yung erythromycin, ito yung clindamycin. So if you would notice, nagfa-flatten yung zone of inhibition dito kasi unti-unting lumalapit yung organism sa clindamycin because they are starting to develop resistance to it. Okay? So, in the event that the D-test is positive, clindamycin must not be given to the patient. Actually, may isang version ng tanong dito. D-test positive daw yung organism. How do we report it? The organism is D-test positive. How do we report it? So, pag D-test positive, okay, pag D-test positive, we report it as erythromycin resistant and clindamycin resistant. Okay? If the organism is D-test positive, we report it as erythromycin resistant and clindamycin resistant. Okay? Next, we have by the way, I just would like to take note, no? Baka lumabas din sa exam, no? The gene responsible for inducible clindamycin resistance is the ERM gene. Okay? This is the gene, no? This is the gene responsible for inducible clindamycin resistance. Okay? Next, we also have vancomycin-resistant enterococci, no? Detection. So, vancomycin resistance, meron tayong dalawa. We have the VAN-A mediated and we have the VAN-B chromosomal mediated. So, VAN-A mediated, inducible high-level resistance to vancomycin and teicoplanin. Whereas VAN-B chromosomal mediated, okay, variable levels of vancomycin resistance and susceptibility to teicoplanin. Okay, so molecular assays that are PCR-based are used to detect VRE. And the most common vancomycin-resistant species is your Enterococcus faecium. Okay, then we have ESBL detection. How do we do this? The dis-agar diffusion test uses indicator drugs, no, for us to be able to detect ESBL. So sino daw itong indicator drugs na ito? We have cefotaxime, okay, cefotaxime and ceftazidime, no? And ceftazidime. Okay? On separate discs and also discs that contain both the indicator drug and clavulinic acid. Guys, tandaan na si clavulinic acid is a beta-lactamase inhibitor. Okay, so how do we interpret the result here? If the combined disc zones, if the combined disc zone sizes are larger than either disc with indicator drug alone, ESBL is produced by the organism. Okay? So if the combined, no, if the combined disc zone sizes are larger than either disc with indicator drug alone, ESBL is produced by the organism. Okay? Kasi yung punto lang naman dito, no, let's say for example, if the organism is a beta-lactamase producer, magiging mababa yung kanyang, magiging narrow yung kanyang zone of inhibition for the indicator drugs. However, if nilagyan mo ng clavulinic acid, lalaki yung zone of inhibition. Why? Because there is clavulinic acid that inhibited the beta-lactamase. So, if the zone of inhibition for the drug, okay, if the zone of inhibition for the drug is larger, no, than the indicator drug alone, then ESBL is present. Okay? So pag ito, para simplihan, pag indicator drug alone, no, indicator drug alone, mas mataas yung kanyang zone of inhibition as compared to the indicator drug clavulinic acid plus clavulanic acid pala. Okay? Ibig sabihin, positive siya for ESBL. No? If the zone of inhibition is wider than the zone of inhibition of the combination of the indicator drug and clavulanic acid, then ESBL is present. Okay? And lastly, we have the Hodge test. Okay? So the Hodge test naman, guys, is by the way, please take note also that the resistance responsible for ESBL is located in the plasmid gene. Okay? Resistance okay, in the plasmid gene. Okay? And lastly, we have the Hodge test. So the Hodge test uses this diffusion method in which Escherichia coli ATCC strain suspension is spread over the MH agar. Then a single imipenem disc is placed on the center of the agar. The organism being tested is then inoculated. Is then inoculated to MH plate by drawing lines on top of E. coli suspension from the edge of the imipenem disc to agar's plate edge. So gaganyan ka, magsislick ka ng paganyan, kumbaga. Okay? So an organism producing carbapenemase will hydrolyze the imipenem and the E. coli will grow at the edge of the imipenem, this producing a clover leaf-like indentation. So for example, no, itong organism na ito, let's say this is our positive control, no? Itong number 3, itong number 3, let's say that is our positive control. Then itong number 2, this is our negative control. Then itong number 1, this is our test organism. So makikita natin dito that the test organism is carbapenemase producing. Why? Because the E. coli here is slowly growing towards the imipenem. That is because in this area, no, the imipenem is already hydrolyzed, no? Same goes with the positive control. The positive control, of course, is an organism that is carbapenemase producing. Kaya unti-unting lumalapit yung E. coli kasi nahahydrolyze na yung imipenem sa area na ito. Kaya sila lumalapit. Dito, hindi lumalapit yung negative control because hindi naman carbapenemase producing yung organism na ito. Okay? So, sa ASCP, no, lumabas itong Hodge test na ito. Yung tanong is, meron daw person, no, with pneumonia. Then the organism causing his pneumonia was found out to be Hodge test positive. Okay, modified Hodge test positive. Ano daw yung antibiotic na bawal ibigay? Yung sagot doon, imipenem, no? Hindi mo siya pwedeng bigyan ng any carbapenem. Why? Again, wala siyang silbi because the organism is a carbapenemase producing organism and it will just render any carbapenem inactive. Okay? Now, sino naman yung gene na responsible for the carbapenem resistance? We have the BLA KPC gene. No? The BLA KPC gene. Okay? So if you have this, you are resistant. Diba? Remember that carbapenem is a beta-lactam, no? So you are resistant, okay, to all, okay, beta-lactam antibiotics. Okay? So I think that would be all for this session. Let's continue with the different organisms, no, in our next sessions. Maraming salamat.